This modified ATAC-seq protocol introduces a new lysis buffer that decreases contaminating mitochondrial reads from 50% to less than 3%. The decreased mitochondrial contamination will allow researchers to perform more efficient ATAC-seq with a 50% decrease in sequencing costs. We have validated this new ATAC-seq protocol in human primary PBMCs, human primary monocytes, mouse, dendritic cells, and melanoma cell lines. We believe it will be effective in a wide range of cell types.
Minimizing sample loss during the cell activation and selection process is critical for obtaining high-quality results with ATAC-seq. We will demonstrate how to handle small numbers of cells from precious samples using basic laboratory equipment.
To begin, thaw 1 vial of previously isolated CD4-positive T cells in a 37 degrees Celsius water bath until the ice has just melted. Then gently transfer the cells to a 15 milliliter conical tube containing 9 milliliters of pre-warmed complete RPMI. Centrifuge the cells at 1500 times G and 4 degrees Celsius for 6 minutes. Then discard the supernatant and gently resuspend the pellet in 2 milliliters of complete RPMI. After repeating the centrifugation, discard the supernatant and gently resuspend the cells in 0.5 milliliters of complete RPMI. Use a hemocytometer to count the cells. Using complete RPMI, adjust the cell suspension density to plate 50,000 CD4-positive cells in 200 microliters per well of a 96-well round-bottom plate. Then, to prepare magnetic beads, aliquot 12.5 microliters of human T activator CD3.